All right, welcome everyone to PDA DACH chapter's webinar on visual inspection. We are very excited to have you with us today. We're going to start right away. Let me give you a quick introduction of our speakers today. I want to start with Roman. Roman is the CEO of Clear Solutions Laboratories. He has extensive experience in drug product manufacturing as well as development. And before Clear Solutions, Roman was with Roche and Lonza. Elizabeth is the Senior Lead Visual Inspection at CSL Behring. She's been leading a global program for harmonization and application of best practices in visual inspection across the CSL network since 2017. Marcus is the product owner of visual inspection software at Kerber Pharma Inspection. He's using his 15 years of experience to actively drive innovation in automated visual inspection. And my name is Antonio. I'm the Global Head of Visual Inspection and Particle Lifecycle Management at Takeda. I've been with the company for 15 years, with the first 10 years spent in manufacturing and the past 5 in global quality in my current role. So that's your lineup for today. Let's jump right into the agenda. I'm going to spend the next couple of minutes to give you a quick introduction about the PDA and the PDA DACH chapter. But then I'll hand it right over to our first speaker, Roman, who's going to run you through the regulatory framework for visual inspection. Elizabeth will pick it up from there to give you an overview of the training and qualification requirements for manual visual inspection processes and inspectors. And Marcus is going to wrap it up with his presentation about inspection technology. So he's going to cover semi-automated and automated visual inspection technology. At the end of this webinar, we're going to have a Q&A session, and I want to highlight one thing. If you look at the menu bar of your MS Teams application, you will find a little button that says Q&A. Hit that button and you get a chance to actively post questions throughout the duration of this webinar. So whenever a question comes up, feel free to enter it in this mask. You will also see questions posted by other participants. And there's a little arrow on the left-hand side of each question that points upwards. So if you hit that arrow, you actively cast a vote for this question. So the more votes a question gets, the higher it will be ranked in the priority. And we're going to focus on the top priority, the questions with the highest rank at the beginning of the Q&A. So make sure to cast your vote for those questions that you feel most interested in, and we'll make sure to have those covered for you at the Q&A. All right, let's give a quick overview about the PDA. What is the PDA actually? So the Perennial Drug Association is a global community. It brings together professionals from across the industry to actively drive collaboration and innovation through various volunteer opportunities. So the PDA is actually member-led. However, it's also mission-driven, and the mission is very simple. We want to advance pharma manufacturing, science, and regulation to better serve patients. The PDA DACH chapter is one of 24 regional branches of the PDA. The DACH chapter focuses on Germany, Austria, and Switzerland. And what we want to do is really to actively promote open knowledge exchange in the pharmaceutical industry by connecting manufacturers, suppliers, and regulatory authorities to really drive this open and comprehensive knowledge exchange. And all that in order to strengthen the DACH region as an industry hub. So if you're interested in learning more about the PDA DACH chapter, feel free to hit one of those links and get in touch with us. Maybe you're up for one of those great volunteer opportunities and want to support our mission. We'll be happy to touch base with you. So since our webinar today is focused on visual inspection, I really can't miss the opportunity to highlight that there is an actual PDA visual conference coming up in the next few days. So we're going to meet in Raleigh, North Carolina on March 10th and 11th. And we're going to really do a deep dive into all the regulatory requirements, the latest technological advancements, and also the best practices that have been successfully applied across the industry. So there's a lot to take away and bring back to your facility and really boost your visual inspection processes. Along with the conference, we also offer training courses. So there will be one training course, a more general one, focusing on visual inspection as a whole, and a specific one focusing on automated visual inspection. Those are right before and right after the conference. So if you're interested in either visiting the conference or joining one of those training courses, feel free to follow that QR code that you see up on the screen. It will give you all the details. I also want to highlight that PDA week is coming up. Beginning of April, we're going to meet in Palm Springs, California for the PDA week that was previously known as the PDA annual conference. So this conference is not only bringing together like all the experts from the industry across all disciplines, it's not only focusing on like GMP manufacturing, regulations, regulatory aspects. I'm bringing this up today because it has a specific session that focuses on visual inspection. So if you happen to be around in California, beginning of April, we'd be happy to meet you there and talk about visual inspection. One more thing I want to highlight are the PDA interest groups. Think of the PDA interest groups as sort of communities of practice. So we actually bring together all the experts from across the industry to tackle common challenges together. There is a specific interest group that focuses on visual inspection. And if you're a PDA member, it only takes you like 3 clicks to join this interest group. The interest groups routinely meet on a virtual basis. However, there are also in-person events that they host on an infrequent basis. So if you want to stay up to date and make sure that you don't miss out on any updates or changes in the realm of visual inspection, I highly recommend joining that interest group. Go to pda.org and follow these instructions on the screen. You're not going to be disappointed. All right, we offered this webinar today as a free webinar, so there is no requirement to be an active PDA member to join the webinar today. That also brings a lot of non-PDA members to the meeting today, and we thought about, could we do something special for you? So we came up with this 20% discount code. 20 discount code will actually give you 20% on your PDA membership if you decide to join the PDA. And I cannot emphasize enough how valuable it is to be a PDA member. It really gets you connected with all the experts across the industry. And also, if you want to join conferences or trainings offered by the PDA, it gives you a significant discount if you're a PDA member. And also, it gives you access to those very, very valuable materials like the technical reports and best practices and surveys that the PDA performs. So really a very, very good deal. If you want to join, you can follow this QR code right on this screen. However, if you want to join at a later point in time, make sure to take a note of this discount code. All right. Without further ado, I want to hand it over to our first speaker, Roman. Roman, can you navigate us through this regulatory landscape for visual inspection?
Also, a very warm welcome from my side. And before I start, I want to take the chance first, thank Antonio. I think his engagement and motivation here is phenomenal. The service he does for the visual inspection community globally can't be underestimated. And also the complete PDA DACH chapter leadership team. I'm very happy that they gave this important topic a stage. I started my career at one of the larger biopharma corporates here in Basel. And compared to the highly complex bioprocess, visual inspection always had a bit the reputation of being non-scientific, trivial, simple, and of course by this never allowed to fail. And I think this is quite surprising because if you dive deeper, the complexity somehow unfolds. Many questions remain unsolved. And I more or less know not a single company who doesn't have any challenges with visual inspection or particle control. Of course, to a different extent, some have very serious issues with visible particles, for example, audit inspection findings, challenges to release batches, or delayed filings, and others are just having questions, how can I set up a globally accepted visual inspection strategy? So my wish for this presentation and also for the whole webinar is to somehow shed a general introduction to the topic and then also set the stage for Markus and Elizabeth, which come later after me, and to also foster the dialogue in the Q&A. I can also only recommend you to join the PDA DACH chapter. I think it's the perfect platform to connect and exchange for these technical topics. And I have the feeling like a lot of great things will come over the PDA DACH chapter. So let's jump right in. Why do we inspect? Visual inspection typically has a dual function. It's an essential process of the pharmaceutical manufacturing process, but also quality control. On the one hand, it has the purpose on ensuring product quality, patient safety risk, and then on the other hand, ensuring GMP process control. Every unit is inspected for known and unknown defects. So I think it's also a great tool to build process know-how. And it's very often a starting point for operational excellence initiatives in the manufacturing process. And what we've also seen in the past years that visual inspection is almost always a focus topic for quality audits and inspections, and very often related to critical findings. So if we look at the top findings, particles or visual inspection is always in the top findings of every year. And just as an example, I brought you two. And of course, there are many cases about the traditional, classic topics about the visual inspection method, operator qualifications. So I selected two which somehow illustrate the increasing expectations by health authorities. So the first one is visual inspection for cell therapy. So in the past, cell therapies were quite often launched and filed without any testing for subvisible and visible particles. So they just simply got waived. Or if something was done, it was not to the extent we know here from normal pharma operations. And for time reasons, let's focus here on point B. So this is about a cell therapy product in bags. Your procedure for removing particles detected in the final product does not provide assurance that all particles, including particles that are not easily visible, can be identified and removed. So you see this has changed. There's now a clear expectation by the FDA, and we've also, by the way, seen cases from European agencies that even though it's challenging to inspect a cell therapy product which is turbid, there is an expectation that an efficient visual inspection program is enrolled. Then second, if we look at the second case down here, 2 lots and 3 lots were out of specifications for appearance due to the presence of particles and were not adequately investigated. So this is one example showing that the agencies are moving away from a pure focus of the visual inspection method to the expectation of a quality risk management end to end. So basically controlling every stage of the particle life cycle up to adequate investigations followed by potential operational excellent initiatives. Again, highlighting, ensuring patient safety and also ensuring GMP process control. And I've mentioned in the introduction that many questions actually remain, even though it sounds and seems simple visual inspection. So for example, how do visible particles rate to product safety? What does essentially or practically free of visible particles mean? How do I implement an AVI system? I think this is a very hot question, especially in the light of GLP-1 at the moment. Or is 0 visible particles a workable acceptance criteria? This one is also critical. Can different types of particles be differentiated in visual inspection? And what's the visibility limit of visible particles? I think especially that last question is such an evergreen. What's the visibility limit of visible particles? And if you now go to Google and search size of visible particles, you'll find numerous of illustrations like this in scientific peer-reviewed articles stating visible particles simply start at the size of 100 to 200 micrometers and larger, and then there are visible particles. However, if you go to the USP, you find language like each final container of a parenteral preparation should be inspected to the extent possible for the presence of observable foreign particulate matter, hereafter termed visible particles. So that's the USP definition. In other words, it's stating a visible particle is a particle which is visible. And I know whenever I say this, it sounds very stupid, but it's something completely different. If you compare the first definition, a visible particle is always 100 micrometer. independent of the container closure system, the product characteristics, the inspection method. Or if you state a visible particle is a particle which is visible to the human naked eye, and it's well understood and logical that a particle must be bigger in a turbid cell therapy product to be visible to the human naked eye, as compared, for example, of a particle in a clear solution in a small pharmaceutical glass vial. So we need a method to define visibility of particles considering your product. And for this, typically these NAP studies are performed. You have a VI test set containing of defects, known defects, and defect-free units. The defects are in different sizes. And then this defect test set is inspected multiple times by a qualified VI operator panel. And if we look now to the left, here's an example of one of these NAP studies. On the x-axis, you can see the size of the defects, and on the y-axis, you can see the probability of detection. And each dot represents now a single defect inspected, for example, by 10 operators 3 times, so 30 times. And then in something like 70% of the cases, this defect was basically identified. And then you can define a threshold. For example, a common one is 70%, and everything above is considered a visual defect or a visible particle. Of note here, the blue dots are defects in a 2R glass vial, and the red dots are the same defects but placed in the 20R vial. And you can see that the red population is shifted to the right, meaning to get the same probability of detection, the particles need to be larger. This makes totally sense because a 20R vial is larger and a larger area needs to be inspected in the same time. So particles need to be larger to come to the same probability of detection. Okay, when we talk about particles, actually it makes sense to also take a look at the 3 types or categories of particles: inherent, intrinsic, and extrinsic. I mean, these categories were first introduced in the US and were then, implemented to some extent also in the EU. Extrinsic particles, foreign to the manufacturing process. So the typical examples in the past were parts of insects. I mean, I'm well aware that we don't have too many insect parts now in our pharmaceutical products, but I think it's a very illustrative example. Then intrinsic, from within the process, for example, manufacturing equipment, container closure system, or inherent, part of the quality target product profile. So an innate product characteristic. This could be particles of a drug delivery system. Some companies have defined protein aggregates or polysorbate degradants as inherent particles. Of note, the EP 517.2 does not have this inherent category. So we find very similar language in the EP 517.2, but the categories seem to be shifted. I mean, of course, there's a lot of controversial discussions ongoing, how aligned these 2 chapters are. And if you ask me personally, I think it's a very missed chance to fully harmonize US and European standards. What is understood, and I think fairly accepted, is that the risk from extrinsic, intrinsic, and inherent particles is decreasing, where extrinsic particles have the highest risk. However, there are some challenges with these generalized safety assessments. So sometimes it's difficult to assign a category. So for example, if you take a fiber could be a protein fiber, could be a fiber shedding from a sterile filter, could be a fiber from a personal clothes of an operator, unsterile. So fiber could be an inherent, intrinsic, or extrinsic particle. And visual inspection operators would need to be trained and qualified to not only detect particles, but also reliably discriminate different categories of particles, which is a challenge. It's not impossible, but it's a challenge. And then to top it all up, some inherent particles, for example, some protein particles or aggregates, may very well have a higher safety risk to some patient populations than some of the intrinsic particles. And these examples, I'm mentioning them because these examples illustrate the necessity for product-specific particle risk assessments. Of course, another challenge is that we don't have controlled clinical studies. So it would be absolutely unethical to inject healthy humans with different types and concentrations of visible particles. So we don't have systematic controlled clinical studies on the safety risk of visible particles. However, we know that the safety is impacted by the route of administration, the composition. Is it, for example, protein particles? Is the protein in the particle chemically modified? The patient group, the dosing, the frequency. So I think it's very clear in the summary, we can say that no clear clinical thresholds for criticality can be given. However, we need to perform product-specific risk assessment, considering all these factors. So after this general introduction, I want to shift a little bit to the global regulatory framework. I've listed for completeness like important guidelines in different areas in the world, US, Europe, Japan. So I don't want to read now through all the titles of the guidelines, but and I think it's also important to mention that they're not fully word by word harmonized, but I think one important bottom line is that all these guidelines mention visual inspection, the requirement for visual inspection must be performed. They give a general framework, but they don't typically provide practical recommendations how to specifically perform visual inspection in your company. And this is also a bit by intent because the agency want to allow flexibility for historically grown company strategies. This leads then to different strategies in different companies, and these strategies need to be justified. I think this is the important part. Your visual inspection strategy needs to be justified. And if we now look into the specific pharmacopoeial chapters of the visual inspection method, we can, for example, compare the EP 219, the USP, and the JP. And I would say these chapters, although they are not officially harmonized over the PDG, so the pharmacopoeial discussion group, like the chapter 788, I would say they are practically harmonized. They give very similar procedures, durations, background, instrumentations, and also suggestions for acceptance criteria. However, of course, the different wordings always leave room for some nitpicking. For example, the USP, just one example, the USP states for the illumination larger than 2000 to 3750, and other chapters may not give this wording. So again, here, I personally believe it's a missed opportunity that we're not fully harmonized over the PDG. Of note, some other nations can provide you with, for example, the Chinese pharmacopoeia can provide you with different procedures and different acceptance criteria. This needs to be considered. Okay, before I close down now, I want to quickly talk about the visual inspection process. And I'm sure most of the webinar visitors here have seen a flow chart like this before, or a similar one, which you can see in the USP 11790. After the filling, we have a 100% visual inspection as part of the manufacturing process, followed by statistical AQL sampling. An interesting aspect, I've shown a similar illustration 7 years ago, and we just came back from a survey of European large pharma companies. And back then, a lot of the companies did not perform AQL testing. Now in 2025, I think AQL sampling has become an industry standard. AQL testing can fail and can be repeated, but it needs strict rules and predefined measurements anchored in the quality system, how and when a repeat can be done. And typically includes that the defects need to be randomly occurring and not suggesting a complete system failure, loss of sterility. If this is the case, typically the batch is rejected and investigation is started. A lot of companies leverage then the AQL results for their QC release. Some do some small sample subsets on top, and then the batch is released. I think an interesting aspect, which is sometimes overlooked, is the QC stability strategy. And here, company strategies vary again. So, of course, because of the small sample set on stability time points, the statistical considerations are not very meaningful. So some companies allow 0 defects if they test then 10 to 20 vials. Other companies allow a few defects and have then a translation table, how this result translates into practically free of particles. Some companies pre-inspect the stability batch because they only want to see the stability trend. Others inspect on enhanced condition before they start the stability studies. And I'm giving these examples again to illustrate that the regulatory framework gives a general frame, but then in each box there's many details which needs to be defined on a sponsor-specific basis. So pharma companies needs to define each and every step, and they need to justify this. There is flexibility, and this is why different pharma companies have different strategies. This is absolutely okay. It simply needs to be justified. So how can we bring this all together? A holistic strategy. On the one hand, we want to ensure, again, patient safety. On the other hand, we want to ensure process control, GMP process control. It starts with quality risk management, so which has a foundation in product and process know-how. Quality needs to be built into the product, can't be achieved by pure testing. This starts with formulation, container closure system, facility equipment, single-use components, and so on and so forth. We have talked about product-specific particle risk assessments to ensure patient safety. The inspection process, it's not just a visual inspection method, it needs a harmonized global process for a global manufacturing network, including visual inspection training, qualification, test sets, visual inspection method development, up to facility material libraries for investigations. And in terms of practical implementations of VI in routine manufacturing, some additional concepts and aspects I think need to be considered. For example, trending, pre-established alert limits, predefined procedures for 100% inspections, AQL sampling strategy, also including the important QC stability piece, which we've just discussed, and then a harmonized investigation strategy. Typically, this is a staged approach, and the investigation should be linked to toxicological inputs. So risk tool is required there. And with this, I already want to summarize and then hand over to Elizabeth. All injectable drug products must be visually inspected. It's a regulatory requirement. It's a very critical component of the quality system. Visual inspection is a probabilistic process, so we need to define visibility of defects on a product-specific basis. This is extremely important for training and qualification test sets, but also how to set up an automated visual inspection system, and a robust and holistic strategy needs to be implemented. And with this, I want to hand over to Elizabeth.
Thank you, Roman. So I also want to thank you for the opportunity to present this topic today. I've spent now 7.5 years at CSL Behring in different visual inspection roles, and I've seen definitely changes in regulatory requirements, what guidelines are available, and also with the collaboration between the other sides within our network, but also within the tech transfer projects we have internally and externally. I've seen different approaches, how operators are trained and qualified, and I would like to present to you today, let's say, the best practice or the condensed version, how to train and qualify operators for visual inspection. So typically we start with our manual visual inspection process flow. So how can we generally qualify new operators in this area? And there are some prerequisites. So not everyone can work in visual inspection. Not everyone is suited for this kind of job. The operators have to run through a specific theoretical training where they are introduced to visual inspection general procedures. They learn how to correctly perform the manual visual inspection sequence of containers. They have a practical training period, and only after passing training and practical and theoretical, they can proceed with qualification. The qualification is always product specific on specific test sets, and if your company produces different products in small vials, big vials, different containers, maybe lyo and liquid products, different syringe types, you should be really careful of describing your bracketing approach in your site procedures, because this is definitely a point auditors are looking into it. So the prerequisites for manual visual inspection operators in general are they have to pass a visual inspection acuity eye test, so they have to have 20/20 vision, and if this is not possible, then they have to wear lenses or prescription glasses, and they have to pass a color perception test. The reason is that the operator should be able to distinguish between the colored product solution and particles, or different colors of particles. And if you're not able to do this, they're not suited for this job. So the results of this assessment are stored in the medical folder of each visual inspection operator, and there's a annual reassessment of vision acuity. And we should also be careful of that there's a different requirement for products distributed to China. The visual test has then been performed each 6 months, so on a shorter period than compared to other markets. We'll then proceed with a theoretical training of the operator, and typically this is guided through a learning management system. So procedures and training packages are assigned to a new operator, and they have to train on specific items within visual inspection. So how to gown and be careful of housekeeping. So they have to be aware of how to clean equipment, the surroundings, the work in. Safety is a very important aspect. So visual inspection operators need to wear protective glasses and safety shoes, and also gloves during visual inspection. Very important is that new operators are trained how they fill out batch records, what are the GMP principles for proper documentation, and what we always think of is reconciliation, so the amount of units that are inspected, and line clearance prior and after each batch. Typically, each company should have a defect library, and this is typically at least a defect catalog with pictures representative of the manufacturing, or the defects that can occur from manufacturing, from different lines and from different products. And ideally, this defect library is also present in a training test set, so the operators are able to not only look at pictures, but only to see real defects in reality. The operators have also to be trained on what is a minor, what is a major, what is a critical defect. So they should be able to distinguish them then in routine. And I want also to emphasize that there should be no reason to rush through manual visual inspection process or through operator qualification, because this is your foundation. If you don't train your operators properly, there's really a high risk in the routine inspection. So we emphasize that the operators have to train the inspection sequence, how to inspect different products and different sizes of containers at the manual inspection booth. And they also familiarize themselves with the training test set. And after this theoretical training, there's also practical training period, because it's like with driving a car. You can't just look into the theory and then start driving a car immediately. This is not possible. So you have to train on real products or train with training material. And if the operators are trained with routine product, then this is always happening under supervision. So a supervisor is always nearby, and the inspected units from the trainee are then verified. And for example, missed defects are discussed with the new operator. This is a very important phase of the qualification or training and qualification phase, because the operator should have enough time to familiarize with the product portfolio and be really confident in doing this job. If the training period is over and passed, then we can proceed with the practical qualification. And this is then happening with qualification test sets. But before this, I really want to emphasize the manual inspection sequence. So this is typically described in the pharmacopoeias. You have to inspect your product against the black and white background for a specific time. That's it. There's really much more behind this, because you have to consider what kind of containers do you have on each side. It's really a difference if you inspect just one unit at a time, or if you inspect multiple units, let's say 10 syringes at a time, or if you have 2 to 4 bottles in your hand. And you have to ensure that with your proper inspection technique, with static inspection, swirling or inverting of the container, you are able to identify not only container defects, like cracks in glass, for example, or issues with crimp caps, closure stopper, but you should be also able to identify wrong fill volume, like underfilled or overfilled units, discoloration within a batch, turbidity, and of course, as mentioned already before, particles and fibers. And if you don't train your inspection sequence properly, you will miss those defects in routine. If your company uses multi-unit inspection, for example, some tools like a clip or a clamp to hold several units at a time, this method has to be qualified. So I will now proceed with the threshold studies. This is really a basis requirement. There are also some other formulations of wording for this on the market. This could be also a probability of detection study or a baseline study. It's all the same. So you just want to know your process capability by doing this. And the intent is that you compile a threshold test set. It consists of all possible defects, and critical major and minor defects have to be in. Not more than 10% defects should be in, like in routine qualification test sets. And the threshold test study is needed to really identify what are your operators capable of identifying and detecting under routine conditions. For example, you would maybe, if you have difficulty to inspect products or colored solution, you would not find 50 micron particles in there. This would be definitely higher. It's very important to say that the threshold study itself, it's not only focusing on particles, because you also would find other defects in routine, not only particles, and you want to know what your operators can achieve in the routine. And typically, the study is done within 30 to 50 runs. You could also have less runs, but this needs to be justified. And coming from what you have to consider for these test sets to know your process capability, that the defective units you use in these threshold test sets are hopefully not only artificially made, but also representative of routine manufacturing defects. Real product should be used because you want to see particles or particle behavior in the real product, and operators should be trained and qualified with real product. And very important is when you perform this study that you consider different particle sizes. Here it's just an example to start with 50 and go up to 5,000 microns. I know this sounds really massive, but for some specific products, you will find particles or fibers in very high ranges. And what is also important is to consider enough samples to put into this threshold study test set, because this is your basis for operator qualification test sets. And you also want to have sufficient samples for future and also spare parts for replacement. What you need to consider is your qualification test sets. They have to be qualified prior use, and they have to be approved by quality. So typically this is a quality department related to visual inspection, like shop floor QA. And there's also one interesting item that I wanted to discuss or present to you. There's sometimes when you have new facilities or you have tech transfer projects and you have an issue, do you need qualified operators first, or do you need a qualified test set first? What happens if you don't have the one or the other? So a good approach would be that, for example, if you have 20 new operators and you want to qualify them, you could compile a test set with possible defects in, as I said before, with critical, major, and minor defects. Also consider the 10% defect rate, and take all the information you have, for example, from upstream departments. What are the product characteristics? What kind of defects could come in via the filling lines or via the primary packaging materials? Then you could run this initial threshold test or study to get like the preliminary results or detection rates for your new operators. And then you could remove, for example, the high performer and the low performer. And from the remaining detection rates, you could pick up those samples that are within the reject zone and which are suitable to put into a qualification kit. And once you have this qualification test set, your new operators can be qualified with this established qualification test set. And you have achieved two things. First, you have now a baseline where you can see your operators are now qualified, and you have also qualified samples. So after a specific time, for example, after 1 year in production, you could do a re-evaluation and check if the content of this initially established test set is still valid, or you could exchange some defect types and re-qualify again. Very important is that the qualification test sets are all blinded, so that operators are not able to distinguish between what is a defective unit, what is a good unit, and they have to be verified prior and after use. For example, if you have scratches or introduced other defects than the initial one, you should replace those items before you go into qualification. What are now the qualification requirements? This is also taken from USP 1790, which is in general a good guidance for visual inspection. The operators have to qualify on 3 representative test set or from 3 runs, and the false reject rate should be equal or below 5%. So for example, if you have a test set that consists of 500 units, the operator is allowed to sort out 25 acceptable units. The reason why we apply this criteria is that we want also to test if the operator is able to sort out only real rejects. And as I mentioned before, you have maybe to consider also what is produced on your site. If you have different vial types with a volume of 200 to 500 milliliter, or different syringes or different product characteristics, this should be justified in the document how the operators are qualified and with which qualification test sets. And the qualification should be performed under routine conditions and should consider also all operational shifts that you have on site. Fatigue is always a topic, so operators should be also able to perform towards the end of a shift. The requalification itself, it's done annually, and also here with respective test sets. Could happen that you have to perform a requalification maybe in between of this interval because you have changed your criteria or you introduced new defects. But typically the requalification only requires one successful run. So now, what happens if an operator fails the requalification? You should have a procedure for that established, and this should be quality approved. Typically, when the operator fails this requalification, then there's a possibility for a second attempt, but it's never on the same day, earliest on the next working day. And this could be repeated up to 2 times. But if the operator fails again, a retraining is necessary, and the last and final third attempt, if this is failed again, the operator is definitely disqualified from visual inspection activities. Now I'm already at the conclusion slide. So in general, operator qualification, the manual visual inspection itself is like the basis for the inspection of your containers. And it's expected and important that you know what kind of defect types, sizes, and particles your operators are capable of detecting. And this is also the basis how you set up your qualification test set and the basis for further equipment qualification, like semi-automated or automated machines. And with that, I will hand over to Markus for the next topic.
Hello, everybody. Thank you, Elizabeth. And also from my side, big thank you to PDA DACH chapter for the opportunity to present here. And a special thank you to Antonio for driving this important topic. I will give you now a general overview about semi-automated visual inspection machines and fully automated visual inspection machines. I will start with an overview of both the systems, followed by the typical qualification approaches. Then how can you select the right method for your process? And finally, I will show you 2 strategies to improve your inspection performance First, the implementation of a two-stage inspection process, and second, how can AI improve your visual inspection process? So let's start with the semi-automated visual inspection machine. I call it SAVI. The containers will be automatically transported through the machine, and an operator will inspect the containers during the transportation. In the inspection zone, the operator can mark the defect units, and the operator will be supported with special light setup, mirrors from the bottom and the top, and a dedicated rotation profile for the product. This ensures a 100% inspection of the units. In general, the performance of detection is comparable with the MVI and due to the control environment consistent. What are the challenges here? We have a human dependency. We have detection limitations, especially for difficult to inspect products. And of course, the limited throughput compared to the AVI. With the automated visual inspection machine, I call it AVI, you have a 100% inspection without human dependency. The containers will be automatically transported through the entire machine, and different camera stations or other inspection methods like HVLD detecting the defects from your manufacturing process. This method is very efficient and provide a very high throughput with a consistent high quality. Also, the overall reject zone efficiency is normally better compared to the other methods due to a smart combination of the light setup, rotation profile, and inspection algorithm. What are the challenges here? You have a higher risk for false rejects, especially during the ramp-up phase, and the effort to develop a recipe and qualify it is significantly higher compared to other visual inspection methods. Let's have a look now on the typical qualification approaches of these two systems. On the left side, you see the high-level workflow for the AVI. First, you have to develop a vision recipe. For this part, you need a dedicated test kit with good units and defect units. And normally, the recipe will be created from an AVI expert. This could be our internal SME, but also our expert from the machine vendor is a common approach. Then you can do your qualification run on the machine. This is normally done with a certain number of test runs with a test kit. The goal is here that your inspection process is equal or better as the MVI. And after successful recipe qualification, you can do your process qualification, typically done with 3 routine batches with extended sampling. After the successful runs and passing the acceptance criteria, you can release the recipe for routine production. On the right, you see the workflow for the qualification of the semi-automated inspection machine. This process consists of 2 parts. First is to train the operators on the machine and also with a test kit. And second is the operator qualification on the semi-automated machine. The goal is here similar as with the AVI. You should reach the same efficiency as the MVI. After successful qualification of the operator, you can start with the routine production. Now you saw different visual inspection methods. The question is now, how can you select the right inspection method for your manufacturing process? So the AVI is a very fast method, up to 1,000 units per minute. It's very efficient and a consistent method. AVI is more efficient as the MVI, but compared to the AVI, much slower, typically 25 units per minute in production. You have a smaller footprint compared to the AVI, and you have a high flexibility through the human inspection. That means the qualification effort for new products are lower. The MVI is very flexible to adapt, and you can use bracketing approaches for your qualification. And the decision is based on experience. You also have directly classification of the defects, which helps to understand the process and gain product knowledge. So as you can see, The decision of which method is the right one depends on various aspects. At least you should select the method based on production volume, diversity, and investment costs. For example, if you have a high volume drug product in just one presentation, a AVI method could be the best solution for your process. As I mentioned before, one of the challenges in the AVI is the higher risk of false rejects. The question is, how can you mitigate the risk? And one strategy is the implementation of a two-stage inspection process. In this process, you combine the AVI method with a second inspection. Typically, you would use SAVI or MVI in the second stage. And as you see in this workflow, in the first stage, critical rejects like cracks will be directly sorted out, and potential ejects, that means real rejects and false rejects, will undergo in a second stage inspection, and this second stage takes the final decision if it's a real defect or not. It is recommended to have here for each goods stream a separate AQL, and after successful AQL, you can combine the streams to one bulk for the next process step. You can also use the same method in the second stage, but you need here a strong justification. So for example, a justification could be you need additional holding time that air bubbles can disappear. How can you qualify a two-stage inspection process? You have to qualify each systems like described above, and then calculate the theoretically overall reject efficiency over the whole two-stage approach. That means you have to multiply the two PODs and compare it against the MVI. And I want to highlight here that the overall POD becomes always worse if one of the PODs from the stages is lower than 100%. But if you can show that your two-stage approach is efficient and you succeed the predefined acceptance criteria, this approach can significantly reduce your false reject rate. So the key takeaways for introducing a two-stage approach: you should clearly understand and document the limitation of the first inspection and why you do a second stage. Just to say to reduce the false reject rate might be not enough. Using the same method, AVI followed by AVI, is not recommended. If needed, consider that you have a strong justification. Consider that each stage decreases the overall POD of defects. This needs to be assessed during the qualification. And the last point, a dedicated extended AQL for stage 2 is recommended to ensure that the process is working with a potential higher population of defects. Another option to reduce the false reject rate is to use an AI-based algorithm. One key benefit is here the ability to differentiate between similar patterns. That means a trained AI model can distinguish between air bubble and particle in a reliable way. On this example, you can see two images, each processed by the same AI model. The left image is a particle defect, a glass particle which is laying on the plunger, and the right image is a good unit with an air bubble on the plunger. On the lower images, you see both heat maps. A heat map is a common tool to visualize how the model is making a decision. And the red area in the heat map shows that there is a high activation for defect, and this is probably the reason for the reject. And in our example, on the left image, it is clear visible the red area covering the particle, and together with a high defect prediction score, it is classified as a defect. On the right image, there is no red area over the air bubble, but some activation appears around it. But together with a low defect prediction score, it is not classified as a defect. With classical vision rule-based algorithm, this inspection job would be more challenging. So I want to illustrate the main advantage of an AI-based algorithm compared to a classical computer vision algorithm on this slide. The black line indicates the fixed threshold of a classical vision algorithm. So you have no chance for clear separation of all real defects and real good samples from your production. You will always have a number of units which are in the gray zone. So you have to find a compromise between a sufficient detection rate and a low false reject rate. And this could be a challenging and continuous task. And the yellow line in the graphic demonstrates the separation of good and defects of an AI-based algorithm. How can you read this line? The AI-based algorithm is looking for patterns and balanced learned combination of rules that you could say the rules are flexible based on the current case. So you see here a better separation of real, good, and defect units. So what is the key message here? With using AI in automated visual inspection, we have the option for overall better inspection performance with less false reject, but also a better detection rate. How can you implement an AI model in your RV process? First, identify your current issue in your production, then specify what the AI model should find and how to integrate it in your vision concept. Which classical parts do you want to keep or remove? Then build a new model. That means labeling, training, and verifying. Also document the model development process, and then freeze and qualify the recipe with the AI model, and then run the AI model in production. Now I come to my last slide with my conclusion. Semi-automated inspection machines are flexible but slower and depends on human decision. Fully automated inspection method is fast and consistent but requires more effort in qualification. A two-stage approach can improve your inspection process if properly justified. And AI in visual inspection bring new possibilities and can improve defect classification and reduce false reject rates. Thank you for your attention, and we're coming now to our Q&A session, and I will hand over to Antonio.
Thank you, Marcus. Thank you, Elisabeth and Roman. Thanks to all the speakers. It was really, really interesting, great presentations there. Let's give them a big round of applause. I already see many hands clapping coming up on the screen. Great job. All right. We are at the Q&A session, and I see there are many, many questions that were posted here in the Q&A function in Teams. I also see that many people have used the upvote functionality, so there are questions which have received a higher priority, and as mentioned before, we're going to focus on these first. Let me start with the first question. I guess on everybody's mind, will the presentations be shared afterwards? Let me answer that with a quick yes. We're going to share the materials. We're going to upload them on our PDA DACH chapter website, and we're going to share that link by email. So everybody who has registered today will receive that email and be able to download the materials afterwards. All right, jumping to the next question, and it's a very, very interesting one. This one is from Dennis. Is there an expectation from regulatory agencies regarding minimum size of visually detectable particles? Is there an expectation that 200 micrometer particles are detectable with probability of detection more than 70% for all particle types and product presentations? And is there a regulatory risk if our NAP study shows, for example, that the smallest detectable particle is 500 micron instead of 200 micron? So maybe, Roman, do you want to give it a start?
I can start. Thank you very much, Dennis, for asking this question. So this is actually a question we here see almost every week. And the answer maybe has two parts. First of all, we have not seen, neither in the FDA audits we had in our own company a couple of years ago or with our clients, that agencies come in and request a 100 or 200 micrometer limit for any presentation, any particle type. So a good scientific justification based on a NAP study was typically satisfying. Of course, there are some extreme cases. For example, I've worked on a product which was a red blood cell bag. their particles only in the centimeter range could be seen. And at some point, you need to define when is a product difficult to be inspected. And also here, no clear rules apply, but at some point, you need to do destructive testing. Some people have given different answers, and there's no clear industry strategy. Some say if the PUD, if the size of the particle is increasing by more than twofold, then it is suggested to start thinking about destructive testing. So for example, John Schabushnik has mentioned this on the last year PDA visual inspection workshop. But there is no clear defined expectation or strategy.
Great answer. Anyone, any more details to add?
I think I can also confirm what Roman said. You can't just say you have to find 200 micron particles if it's impossible with your product. This is just not feasible. And if you can justify what is your capability and what are the limitations, and you can show this to an auditor, this is absolutely sufficient at the moment.
And many details also apply here. So for example, if you look at fibers, a lot of companies measure the size of a fiber by simply measuring the length. And quite often, fibers can only be seen in the range of 500 to 1000 micrometers. And that's absolutely acceptable. So if you use the Ferret Max, for example, if you use ECD measurement of fiber, then smaller fibers can obviously be seen, 100 micrometer fibers. So there's a lot of details, and it's all comes down, as mentioned in my presentation, good scientific justifications. Our experience with FDA, but also with the European agencies, is good scientific justifications always win.
And that is a great aspect to it. Absolutely. Let me just add one more thing. From my point of view, regulators always want us to take responsibility, right? They want us to do our due diligence. So we all face a lot of, very complex products. Talking about, colored solutions, we might have, proteinaceous solutions, we might have flexible containers. So there are many characteristics that come into play when it comes to, potentially difficult to inspect products. But destructive testing is not the only route we can take. So they actually want us, and this has been updated in USB 1790, I think like 2 years ago, that we introduce, additional options on how to increase detectability of defects, in those specific type of products. So you can actually change your inspection method, you can change your inspection technique, you can change even the inspection equipment that you use. So the inspection booth, the light setup, you could use lights from below, you could use backlighting situations to enhance detection of particles. And that is all, it all comes together to doing your due diligence, making sure that you do the best you can in order to ensure reliable detection of defects. And I also agree, there is no cutoff, no specific cutoff that determines, okay, now you have to do this destructive testing. It is a typical thing that you do for lyophilized products, for example, because it's just understood that, you cannot look through the cake. There's no chance to actually detect any particle that might be embedded in the cake. So this is the expectation that you do some additional sampling after you have performed your 100% inspection. And some of those, samples that you draw, you need to reconstitute so that you can actually inspect the liquid solution for presence of particles. Those sample sets are typically rather small, talking about maybe 20 samples, and they will give you an indication whether you are okay with all the other measures that you have in place, with your holistic particle strategy, with your 100% inspection, with your AQL testing, and if in the end also that supplemental testing, the destructive testing passes, you'll be okay to claim that your batch is essentially free of particles. All right, let's jump to the next question. This one is from Mike. What is a typical false reject rate for automated visual inspection? I gotta hand this over to you, Marcus.
Typically, I think it's very difficult to answer. I think it depends. from my experience, I saw a lot of things between less than 1% to 20%. I think it's important that you understand what is the reason for the false recheck, and you can justify it and explain it. And yeah, you have to let's say you have to understand your process and have it under control. Yeah. I would say if the false recheck rate higher than 20%, you should do something, but this is more a business risk maybe. But if you can explain it, then probably makes sense. But maybe a question to Roman or maybe to Antonio from your experience in your environment, also to Elizabeth, is there? What do you say? Because I cannot say there is a 1% is the value you have to. It's the goal, but I think there's a range. Yeah.
There's definitely a range, and Roman, I'll let you answer right away. But I have a follow-up question to you, Marcus. What is like the typical range that your customers ask for when they place a PO?
Typical range that the customer asks always, you want to have less than 1%, or we have to agree in some numbers, but we are always agreeing on defined test kits. So we receive a test kit and the number of good samples, and we provide for this test kit numbers. And this is our acceptance criteria. But later, if we go in production and start up the production, we have more variances, and this is something which is coming to the production tuning or fine tuning, where we have to see what the continuous work, how we can improve the recipe, and yeah, that's yeah. But the expectation from is always 0. But I think all of you know that this is very hard to get, but we try to keep it as low as possible.
Thank you.
Maybe to add a couple of points here, because I think this is also an extremely interesting question. First of all, I think we have this situation that we balance the POD picking up actual defects with the false recheck rate. They always go somehow hand in hand. If you increase sensitivity, often the false recheck rate goes up, and a nice balance needs to be basically defined. What I think is extremely critical is these threshold studies we've talked about. I've seen many cases where either equipment vendor or a pharmaceutical company comes with a defect set showing something like 20, 50 micrometer defects, which no manual visual inspection operator can see. And then they expect the automated visual inspection machine to find these. And in my opinion, visual inspection, the automated machines should represent the manual process. So a well-defined manual visual inspection threshold study and the results of this can smoothen the implementation of an automated visual inspection system. Because if the system only needs to find, for example, 200 or 500 micrometer particles to represent the manual process, the risk for false rechecks and for low PODs, it's much lower. So defining basically the acceptance criteria with a good and broad NAP set is important. And then a third point is, I think in comparison or in contrast to the manual NAP sets, I believe the NAP sets for automated visual inspection should be a bit larger, because we always forget that the human brain, you show them one time a crack or a scratch, and the human brain can conceptualize and recognize scratches and cracks all over the vial in different orientations. However, if you train a machine, you sometimes don't know what makes the machine recognize a scratch, for example, the orientation. So you need variations of these defects to train the system. So I think if we put these things together, that we don't want the automated machine to be much more sensitive than manual operations and have larger NAP sets, often the question of false recheck rate and finding the defects is already solved, in my experience.
Roman, I cannot emphasize enough what you said about, this balancing act that we have to accomplish. I fully 100% agree with what you said. It's really important, to make sure that we understand our capabilities manually. If you use, for example, throughout the phases of an AVI project, starting with like feasibility studies, and you collect samples from routine production and send those to an AVI vendor to run a feasibility study. And then you see that the results are probably not, what you expected. Say you expect a super high, probability of detection for certain defects. It's really important to understand that we need to understand the composition and the performance that we can manually achieve in this test kit. Just because we found it like one time in our routine inspection process doesn't mean it has a reliable probability of detection. So that needs to be ensured. And I've seen, throughout AVI projects, you get to learn a lot about your actual manual inspection process because you need to redefine acceptance criteria. You need to redefine definitions of what is a defect and, what is variation that you might accept. You will realize, digging deeper into this process, that maybe not everything is clearly defined yet, but you need to get there in order to be able to teach the automated system to make the right decision for you. And oftentimes we tend to overtune AVI systems. We tend to expect like perfection or very, very high detection probabilities for even the smallest defects. And of course, then it starts to, meet the point where just typical variation, which is not a defect, which is not considered any imperfection, will then lead to false ejection. And handling false ejection at a high level is just painful. And at some point, you might even face the situation that, if you qualify this process and you say run the process, the AVI process with 40, 50% false rejects, question might come up, how do you consider this a validated process if you're, rejecting 50% of your batch for the wrong reasons? So this is really important to consider as well. All right. Next question is from Eric. Could you give an example how many companies are using AI-based vision in production and how many images for each defect classification were labeled and how they managed images of rare defects?
Question for me, I guess.
Feel free.
No. So I can't give you here our number. I can't say that every new project are working in the evaluation phase if AI makes sense or not. And the minimum is that the hardware is prepared for the future. That means we have a GPU in our vision processor, not our normal CPU, that you have the chance later on to implement AI on your system. And that's currently the challenge. Using AI in the beginning is a challenge because we do not have enough data. We need data from the reproduction. So that's normally the strategy currently, that the companies are buying this option, with the right hardware, and then later on they will collect the data into production, and then they have the chance to implement AI algorithm. Then next question was, and how many images for each defect classification were labeled? I would take this question for the next session because I have, to be honest, I don't know it by heart from our project, but it's something we can deliver. And the other question was how the managed images of rare defects. Currently, there are approaches that we can create artificial images from the defects. This is something we have data scientists, or everybody has this, and this is the approach. If you not have enough defects or not enough defect images, we have the option to create artificial images. This is something we can maybe also discuss in the next session a little bit more in detail.
Thank you, Markus. This again highlights, we need samples. We need, for AVI implementation, we need a ton of samples. We need various representations, as Roman already highlighted, of the different types of defects at various, positions, sizes, etc. We really need this to be able to develop those vision recipes. And when we talk about AI, I think it's really important to understand AI is not going to solve all problems, right? What we really need to start on is using our traditional vision engineering tools that have been very successful, over the past, say, 30 years, and start with that. Start to achieve the best performance we can get without, making it too complicated. Say there are use cases that aren't really made for AI. A very simple example might be fill volume inspection. That is easily, you can program this in like, I don't know, say 10 minutes. But if you start training an AI algorithm to detect fill level differences, you're going to need a lot of images. So that's just the wrong use case for AI. And there are many more examples like this.
Yeah, Antonio, I couldn't agree more to it. This is so true. And maybe a couple of points to add. I personally believe that neither the general best strategy has been found yet or defined, nor like the practical implementations, how many images, so on and so forth. So for example, the strategies how to train an AI model vary. Some companies train it with a large number of defects. Other companies say, well, this is impossible to create so many different defects. We create some digital images of defects, variations of defects to be more efficient. And then other companies, again, train the system with defect-free containers and then let the AI system identify if you have a discrepancy from the good image. So the other way around. So it will be very interesting to see in the future, but I'm very much with Antonio. I mean, classical image analysis is also a digital algorithm, and this AI training is just another, the next expansion of it, but it's not the solution to everything.
Thank you, Roman. So we have another AI question up top. I'm gonna skip it for now and just take another one that we don't focus too much on AI today. This one is from Miliana. Question connected to NAB studies. How does operator experiences or training qualification status impact NAB study results? Should we include operators with different experiences to have statistically more reliable results? Elizabeth, would you want to take this?
Yeah, sure. This is also what we do in routine. So we mix the experience with less experienced operators. We have all kind of representatives that we get proper results. And I can confirm this, yeah.
Can I give you a follow-up question to NAB studies, which really fits perfectly into that? For arranging or NAB study, which is preferable? 3 inspectors, 10 runs each, or 10 inspectors, 3 runs each?
Yeah, I would say you don't always have to perform 30 runs, but this is another topic. And if you have a justification for that, and I would prefer to have 10 inspectors and 3 runs because you have a much better distribution of different experiences, and the operators don't have the test set 10 times in their hand. So this is much better.
Yeah, that sounds great. I tend to agree with you. And maybe one thing that I would like to add to that, you really should have those 3 runs per operator. If you do like 5 operators, 3 runs, 10 operators, 15 operators, everybody should have 3 runs. And it gives you, the quality of data that you need to assess and understand and identify any outliers. The outliers that you discussed in your presentation, Elizabeth. It's really important to be aware of, do we have any exceptional performance that are just not representing like our mean performance? Or do we have any, weaker performance that we also need to exclude from the data set, just to be able to, catch that mean performance of our inspection process. So unfortunately, we are at the end of our webinar. I really would like to, continue discussing all those questions, and there are a ton of additional questions that we didn't have the chance to discuss yet. However, we need to wrap it up today, and I'm gonna just quickly run you through what's coming up next. We're gonna share the materials. As already mentioned, those materials will be available to you. What we will do with all the questions that you have posted today, we're going to collect and analyze those questions, and we're going to make sure to prepare an overview for a follow-up session. The follow-up session that we're planning, we haven't defined the date yet, but it will be later this year, and you will receive an invitation. Everybody who joined today will receive an email with information that we're planning this additional session. The next session will be a little bit a different format. We're not going to focus too much on presentations, but we'll focus a lot on Q&A. And we'll use those questions that you have posted today as input for this follow-up session. And on top of that, we'll also bring in more experts. So we're going to have a larger panel of experts answering all your questions. So let me say thank you for joining the session today. Thank you to our speakers for the great presentations. And with that, we're going to wrap it up for today. And I'm really looking forward to seeing you all again very soon.